DNA. Have you ever heard about DNA? DNA or deoxyribonucleic acid carry genetic material of human and almost all species in the organism. DNA located inside the cell nucleus, and a small amount of it can be found inside the mitochondria. DNA is composed of nucleotides which are arranged in two long strands that form double helix. The nucleotide consists of three components, which are phosphate group, ribose sugar, and also nitrogenous base. Nitrogenous base made up from four chemical bases, which are A for adenine, G for guanine, C for cytosine, and T for thymine. DNA bases pair up with each other, A with T and C with T. This is known as base pair. However, DNA is just one of the cells that can be damaged 10,000 times per day. What are the factors that can contribute to DNA damage? Some of the factors that had been identified include UV light, chemicals, and X-ray. Exposure to those factors can lead to DNA damage that carry genetic instruction in our cell. Fortunately, our cell has their own way in fixing this kind of problem most of the time, and this process is known as DNA repair. DNA repair is vital for all organisms, as without it, spontaneous and environmentally induced mutation can occur. In humans, even minor type of DNA damage can manifest virus type of diseases. There are three general steps in DNA repair. First, protein in the repair system will detect any form of alteration. Then, the abnormalities in DNA that had been identified will be removed by DNA repair system. Then, DNA will be synthesized normally. The action of DNA repair is based on DNA repair system. There are 5 types of DNA repair system. The first one is homologous recombination repair. The second one is non-homologous end joining. The third one is direct repair, followed by mismatch repair, and base and nucleotide excision repair. In this video, we are going to look deeper on DNA mismatch repair. Mismatch. DNA mismatch can be defined as DNA defect when two non-complementary bases are aligned in same pair step of a duplex DNA. Next, we are going to look into how mismatch can occur. Mismatch occurs when DNA double helix disobey the A, T, or G, C pairing. This is due to incorporation of wrong base and failure of the proofreading system. When this kind of problem arises, it will create an incorrect double helix and will be added into growing strands by mistake. This action create a mismatch between parental and newly made strands. But when DNA damage can occur? DNA damage can occur during DNA replication, mutagenic chemicals, ionizing radiation, and also spontaneous deamination. Mechanism of mismatch repair. There are a few components that involve in mismatch repair. The first component involved is DNA polymerase III. Its function is to make DNA from nucleotide, which is the building block of DNA, and then it also plays a role in DNA replication to copy one molecule of double-stranded DNA into two new double-stranded DNA molecules. Next is MutS protein. It functions as a regional lesion sensor that specifically recognizes a mismatch. The next component is MutL protein. MutL protein coordinates the sequential mismatch of repairing steps. Then, it also mediates protein-protein interaction during mismatch recognition. Next is MutH protein. MutH protein will activate the mute exonuclease that cleave the newly synthesized DNA. The last component present is ligase. Ligase will facilitate the joining of DNA strands together by catalyzing the formation of phosphodiester bond. In normal replication of DNA, DNA polymerase will play its role in addition of nucleotide to DNA strand complement to the DNA template. DNA polymerase will arrange bases according to their base pair, which are A with T and C with G. But DNA polymerase also may insert a mismatching of bases where it put, for example, C to pair with T. This situation is known as DNA mismatch. Once DNA mismatch happens, MutS will bind to both DNA strands where the mismatch occurs, followed by MutL. Next, MutL will bind to the nearest DNA sequence that's same to the sequence where DNA mismatch occurs. Then, MutH will bind to the nearest DNA sequence that's same to the sequence where DNA mismatch occurs. MutH will cause double-stranded DNA to curl until MutS, MutL, MutH complex is formed. At this point, exonuclease enzyme will bind to the same place as MutH and start to digest the newly formed DNA strand together with the nucleotides. When it reaches the mismatched nucleotide pair, it will bring the nucleotide away from the DNA template. Then, DNA polymerase III will be stimulated and complement the bases to form into double-stranded DNA. Later on, DNA ligase will join the fragment at where the mismatch point occurs. DNA ligase will later seal the gap present in DNA backbone, and this action will complete the mechanism of mismatch repair.
What is the effect of DNA mismatch? Cancer occurs when cells separate in an uncontrolled way, ignoring normal stop signals and producing a tumor. This bad behavior is caused by accumulated mutations or permanent sequence changes in the cell's DNA.
Actually, there are many types of DNA repair mechanisms that can recognize and remove this type of mismatch.
However, what if other systems fail? If these systems fail, cells contain the additional DNA repair system that can detect base mismatches and fix them. In conclusion, there are interesting DNA repair systems exist in all species of organisms.